Hi everyone, this is Dr. Hinkey and welcome to Bio101. This is Chapter 1, our Intro to Biology. I'm not going to follow exactly the chapter. I'm going to bounce around a little bit, but it will cover the material you'll need to know. The slides are a little bit different from the PowerPoints that are in your D2L classroom. Those guide you right through the chapter as it's presented. Those are from the publisher. Same material, I just added a few different pictures and a different approach. Make it a little more interesting, I think. All right, so biology and the living world. When we are talking about biology, the study of bio, living things. There's incredible diversity in the living world and in living things. Everything from single-celled, simple bacteria and archaea that have no nucleus and have no organelles, have very few structures inside. They're very simple, small, single-celled organisms, through more complex single-celled eukaryotes like amoeba that are protozoans or protists, and then moving up in complexity to sponges, cnidarians, jellyfish, and to different types of worms, to plants, all the way up to animals. This is my dog Muggle. So how do we know that all of those things are living, and how do we differentiate something that is living from something that is inanimate or a non-living, not dead, previously living, but something that doesn't have the capacity for life? So the properties we look for, and we have to have all of these properties, not just one or two of them, but all of them. Life is organized. That means there is structure to it. There is some methodical way that things are put together and organized that we can trace. Living things have metabolism. They have homeostasis. They respond to stimuli. They reproduce, grow, and develop, and they evolve through inheritable adaptations. So let's take a look at these different properties a little more closely. So when we talk about organization, what we're talking about is basically living things are organized sort of like Legos. You can build incredibly complex and elaborate structures from Legos by taking the smallest little single piece with one little nub on top and putting them together in lots and lots of different ways. All living organisms are organized of the same molecules, just put together in different ways. So those levels, what we start with, at the smallest level, on the molecular level, we have atoms, which are made of subatomic particles, protons, neutrons, electrons, that come together to form atoms. Atoms are the most basic, simple form of matter. They can't be broken down into any smaller particles without losing their properties. So an atom is our basic building block. That's our little tiny small Lego that we can put together in lots of different ways. We put atoms together to form molecules. We take 2 atoms of hydrogen, we put it together with 1 atom of oxygen, and we get the molecule water. We take 6 carbon atoms, we put that together with 12 hydrogen atoms and 6 oxygens, and we get glucose, a sugar. We put those molecules together into some bigger, what we call macromolecules, proteins, carbohydrates, lipids, and nucleic acids that form all of the structures within cells, or the organelles, and those we put together to make cells. All this is the molecular to cellular level of organization, because until we get to cells, none of these smaller units, our building blocks, have all of the properties of life, all of those 6 properties that are needed. They have some of them, but not all of them. So cells are our first level of organization that exhibits the property of life. From cells, we can put similar cells together into tissues. So tissues are composed of similar cells carrying out similar functions. We put those together to form organs. So we can have multiple types of tissue. We can have muscle tissue, nerve tissue, epithelium. Put those together to get an organ like a stomach or a heart. And then we put all of the different organs that are involved in a similar function together, and we get organ systems like the digestive system or the circulatory system. And then all of those make up one complex organism or a multicellular organism. We can move on to a broader grouping from those organisms. All similar organisms are called a species, and all of the organisms, all the individuals of the same species living in a specific area are the population. Species don't live in isolation. We live along with other species. All of the different species that live together in the same geographic location are considered the community. And those communities of living organisms don't just interact with each other, but with the environment as well, with the non-living components, the air, the water, the soil. So all of the living organisms, the communities, and the non-living component of an area are called an ecosystem. And all of the ecosystems, all of the different areas that support life on the planet are the biosphere. When we talk about those levels of organization, what we see is this hierarchical structure. And as we move up through the levels of organization, we'll see new properties that emerge. So, for example, hydrogen as an atom is a gas, oxygen as an atom is a gas, but we put those together and we have liquid. So that's a new emerging property that we get. So the whole is greater than the sum of its parts. So cells, the emergent property at the cellular level, is we get those properties that designate something as a living thing. So let's go back and take a look at our properties again. So that's organized. Metabolism. So we hear metabolism a lot when we're talking about diet. Oh, I have a high metabolism. I burn calories really quickly. What metabolism really is in a biological sense is the ability to acquire energy and then to put it into a useful form and use that energy to do work. So we have two parts of metabolism. We have catabolism. That's when we take big molecules and we break them down to smaller molecules that we can then use for energy. As we break bonds, we can get energy, release energy from those molecules. We release energy from our food as we break it down. We also get parts. We get resources to build new biomass. So when we eat protein, that piece of meat doesn't necessarily go to muscle. It gets broken down into the building blocks of proteins, amino acids, that can then get put together in different ways for different structures to do different things. Not necessarily going to be a muscle. It's probably going to end up being an enzyme or another component. But that's the second part of metabolism. That's anabolism. We take those small parts and we put them together to make bigger molecules. So metabolism has both parts, the breaking down to release energy and the building up to get new parts, new structures, new functions that have the needed function for the organism at that time. Homeostasis refers to maintaining a stable internal environment or a stable environment. We talk about homeostasis in biology. We're really talking about physiological mechanisms. These are things that we don't have control over. So if I get cold, I could put a coat on, but that's behavior. That is my response to a stimulus, the stimulus being the cold outside, so I put a coat on. We also have homeostatic or built-in physiological mechanisms that we don't really control. Our body does that to keep us within our operating parameters, our preferred operating parameters, or our happy place. So homeostasis, the most common example that everyone's familiar with is temperature. Our body temperature, 98.6, is our preferred happy place for temperature. If my body starts to get too hot, I perspire, my capillaries start to swell, to open up, to rush more blood to that area, to release heat at my skin surface. All those are mechanisms to bring my temperature back down to its happy place of 98.6. Similarly, if I get too cold, I'll start to shiver, and that motion will generate heat to warm me up. So those are mechanisms in place to keep us in our best operating parameters. Temperature is the most common example, but it's not the only one. Blood sugar. We maintain a pretty steady set range of blood sugar. If we eat a lot of sugar, our cells will release a messenger that says, hey, let's gather up some of that sugar and store it to use later. So this is how insulin and glycogen work. So we have too much sugar in our blood, we're going to store that as glycogen. Too much glucose, we store it as glycogen in the liver. That's a big packaged molecule. And then as my blood sugar drops, I'll break that down back to glucose and release that into the bloodstream for my cells as they need that. So we keep that steady range. We keep our blood, we keep all the parts of our body at a specific pH. It's not the same everywhere. In my blood, that pH is slightly basic, but in my stomach, that pH is very acidic. So I have a preferred pH range where things operate best, and my body maintains that homeostasis. So there are lots and lots of parameters. We'll look at more of these throughout the term, or the operating conditions where we are at our best, and we maintain that internal environment to be fairly stable. Responding to stimulus, it's external, there's stimuli, we respond to the environment around us. Living organisms respond. I get cold, I put a coat on. The sun is bright, I shield my eyes. So we have lots of ways to respond to stimulus. Living organisms reproduce and they grow and develop, have different life stages where we perform differently. So in humans, we start out as an infant and we grow, we develop. We have internal changes that take place throughout our life as we hit puberty and adolescence, and then adulthood and old age. So that grow and develop means how we function changes over time, and that's true of all living organisms. And then the last one is that we evolve through inheritable adaptations. So kind of combined those two. Living organisms have adaptations that allow them to evolve or change over time to best fit into the environment in which they live. We'll talk about that a lot more later in the term. In biology, this term adaptation has a specific meaning, a little bit different from our common use of, oh, I adapt. Well, I moved to South Carolina and I adapted to the heat and humidity. I got used to it. Becoming accustomed to something is not the biological equivalent of adaptations. So adaptations are physical changes, inheritable changes. So they're changes to the genome, to our genetic makeup that we can then pass on to our children. So within a population, there's variation. That variation is due to slight differences in our genes. Some of those variants are better suited to survive and thrive in the environment, meaning they're better able to get food, to use food, to find shelter, to find mates, and to reproduce, have healthy offspring. Others are less adept at that, and so they will pass on their genes at a lower rate. Over time, over generations, those genetic traits, those characteristics that are most beneficial, that allow an organism to survive, thrive, and pass on their genes at a higher rate, means subsequent generations will have more and more members that have those traits. So the population will adapt to where more individuals have those preferred traits. And that change over time is evolution. That happens through natural selection, meaning those best suited to the natural conditions in the environment survive and pass on their genes at a higher rate. So we'll get into that as we study genetics and inheritance in a few weeks. All right, so those are our properties. Because those properties lead to incredible diversity, as adaptations, organisms adapt to the specific niche or habitat they they survive in. And so there are different traits that are beneficial depending on what type of organism you are and where you live. So that diversity, the way we capture that diversity and study that and understand that is through classification. We group organisms based on their similarities. So at the broadest level, that classification is called taxonomy. And at the broadest level, we have domains. So all of the living things in the world can be divided into archaea, which are single-celled organisms that are similar to bacteria, but not exactly, so they have their own domain. Bacteria that are simple cells with no nucleus, as are the archaea, no nucleus, no membrane-bound organelles. But there are some molecular differences that make those two different domain groups. They're significant enough to separate those out. And then the third domain are eukaryotes. So eukaryotes have cell structure. They have a complex cell structure. They have compartmentalized cells with membrane-bound organelles and a nucleus. So those are our three domains. Within those, we can divide those domains into kingdoms. So in the eukaryotes, our kingdoms are protists, which are single-celled organisms like amoebas and paramecium, plants, fungus, and animals. Well, there's a big range within animals. A sponge is an animal. Jellyfish is an animal. Muggle's an animal. So we divide those further into phyla. So we have mammals. We have the phylum Chordata, so chordates, organisms, animals with a backbone. Cnidarians, which include jellyfish and hydrozoans. Porifera, so we have lots of different phyla. These are the sponges. We divide them based on their similarities and on down to class level, order, family, genus, and species. So genus and species together are our most specific. Humans, we are Homo sapiens. I like to consider genus and species sort of like a person's first and last name. Genus would be your last name. Species is the most specific identifier, your first name. But I can't just use species alone, just like I can't use my first name alone. If I introduce myself as Lynn, how do I differentiate me from any other Lynn out there? There are lots and lots of other Lynns. To know exactly who I am, I need to say Lynn Hinkey. So that would be genus, species, Hinkey, Lynn. So we always use these two together. I can use genus separately, so I know I'm a Hinkey. That narrows it down enough that I know where I belong. And then this would tell me exactly which one I am. But we use these two together. We never use species alone because you can actually have lots of organisms in different genuses with the same species name. So we use these two together. The way I properly write genus and species is genus is always capitalized. The species name is never capitalized. And I always write them in italics. Many of you may have been taught that underlining is also acceptable. That convention was really to accommodate typewriters that don't have italics. Now we all have word processors, so we can all italicize genus and species. And if we're writing, we would just write that in cursive. We would not print that. We'd write it in cursive to differentiate it. Once I know that the genus and species that I'm looking at, Homo sapiens, I can abbreviate the genus to just the first letter and say capital H, sapiens. Or back to my analogy of first and last name, Hinkey, Lynn, now I can be H, Lynn, and everybody knows what H I'm talking about because I've established it. So you write it out fully the first time and then abbreviate afterward. Sort of jumping toward the end. Our biology has 4 unifying theories. These are broad theories. We'll talk a little bit about what a theory means, what the actual meaning of that word is when we talk about the scientific method, and we get into that in just a minute. These unifying theories basically areas that we understand on such a level we've found no exceptions. All living organisms obey the rules. These theories give us predictive models. Once I've identified those properties and know something is living, I know they will, I can make predictions about them, and I know how they will work and function because these theories provide a model to tell me how this is going to work. So the cell theory is that all living things are made of cells, single cell or multicellular organisms. All living things, anything that exhibits those 6 properties that we discussed, has cellular structure at its foundation. The gene theory, one gene, one protein, that the functioning, how this cell works, is governed by its genes. And our genes are inheritable. They carry the traits that tell our cells how to work, and those are passed on from generation to generation, which gets into the theory of heredity. The genes are passed on in a very specific manner that we will look at when we get into genetics. How are they passed on? Through meiosis, we make gametes that come together in sexually reproducing organisms, or by making a copy of a genetic material, an exact copy in mitosis for single-celled organisms that reproduce by binary fission. But so the theory of heredity tells me how genes will be passed on, so that I can make predictions about subsequent generations or previous generations. And then the theory of evolution, it tells me that through natural selection, organisms evolve and change over time. So it's change over time so that a population becomes best suited to survive and thrive in the environment in which it lives. All right, so those basically give me some predictive information, some solid foundation from which I can move forward to learn the specifics of how any individual circumstance or a phenomenon is going to play out in the real world if I'm looking at cells. So how I study the real world is using the scientific method. In biology and all the natural sciences, we use the scientific method, which we apply when we have measurable, observable, quantifiable phenomenon that I can repeat, I can replicate to verify my results or to discredit results. So the scientific method, the steps, it always starts with an observation. I see something and it raises a question. So people that look at something and say, that's interesting, and it raises a question, and then they just stop there, not scientific thinking. We're going to raise a question, say, I wonder what that is. What's that about? How does that work? So let's see. I observe that, when I had Oreo cookies for breakfast that day, I had a much better test score. So that would be my observation. My question might be, do Oreo cookies improve my brain power? So that question raised by the observation, I think our book combines these two. It has an observation, and the second step is hypothesis. My hypothesis is not this question. My hypothesis is my proposed answer. So I can't do an experiment and I can't gather evidence to negate a question. A question just asks, what is it? It's the answer that I can demonstrate is wrong or that I can gather evidence in support of. So my hypothesis is my proposed answer to this question. At this point, this is my opinion. This is what I think. It doesn't come out of thin air. It comes from my experience. It comes from my observation, my experience, what I've learned elsewhere through life. We all have experiences, so we can form opinions based on these experiences, on information that I know. It doesn't matter at this point if it's right or wrong. I have no evidence for it. It's an opinion. So we don't want to stop here, because if we stop here and say, gee, here's my proposed answer, that's it, I'm convinced that that's it, we end up looking like a fool. Because what's it based on? Nothing. I pulled it out of my thin air. So Our hypothesis is our starting point. This is where we figure out what it is we're trying to find out so that we can design an experiment to test it. A really good way to state your hypothesis is as an if-then statement, because then it tells you exactly what your experiment has to be. If students eat Oreos for breakfast, then they will perform better in class. I'd make this even more specific and say then they will have higher test grades. Test grades, I can measure that. Perform better in class, that's a kind of fuzzy statement. So I want to make that specific to what it is I'm measuring. They will have better test grades. Now I know exactly what I have to do. If students eat Oreos for breakfast, I need to have students eat Oreos for breakfast. Then they will have higher test scores. All right, what do I have to measure? Test scores. So the components of a scientific experiment include an independent variable and a dependent variable. The independent variable is the phenomenon or the thing you're interested in. How does it impact the dependent variable? The dependent variable, what happens to this part of my hypothesis, depends on what happens here. So Oreo cookies are independent of test grades, right? No matter what my grade is on a test, an Oreo is an Oreo. What does change, what can impact the other variable, is whether or not I eat Oreos. If I eat Oreos, it can impact how I perform in class, how I do on my test. So this is the dependent variable. This is the independent. So the other thing that stating your hypothesis as an if-then statement helps with is identifying these. If I make a statement that says, if students eat Oreos for breakfast, then they will get better test grades. It tells me I. The I in if gives me my independent variable. Then what comes after depends on that if. So this is my dependent or responding variable. And now I know my variables that I'm testing, that I'm measuring. So it tells me what my experiment is. The other thing I need in an experiment, though, is a control group. We think about drug tests with placebos. The group that doesn't get the drug, they get a fake. I need that so I can compare. What would have happened anyway? What would have happened if I didn't eat Oreos for breakfast? So if everybody in the experiment, all the students eat Oreos, then I don't know if the Oreos made them do better or if it was something else that made them do better. Would they have all done better anyway? So what I need is a group that doesn't get this independent variable or doesn't have a change to that. And that is my control group. So in this case, I would have, let's say I have 20 students, I would have 10 of them that eat Oreos for breakfast and 10 that don't eat Oreos for breakfast. And then I could see what impact the Oreos had on test grades. There's one final thing I have to do to make sure that whatever change I see is actually due to Oreos. That is, I have to make sure everything else that goes on with my test subjects is identical. Otherwise, I'd have lots of different variables. So the way you identify these other variables, controlled variables is what we call them. Don't confuse those with controls. The control or control group is what I compare with. The controlled variables, variable means something that can change. So I look here, well, Oreos can change, but what are all the other things that could change that would have an impact on test grades? Well, how much sleep I get, what other food I eat, what instructor I have, what textbook I use, the number of hours I spend studying. All of those things could impact my test grade. So all of those things have to be the same for both groups so that when I am comparing my control group with my test group or my study group, I know I'm comparing the only thing different. I'm comparing what impact Oreos had on those test grades. So all of those other factors that could affect the outcome, that could change my dependent variable, have to be held constant. So I am going to control those variables. So they're called controlled variables. That has two words in it, controlled variables versus just the controls or the control group. So those are easy to confuse, so pay attention to that terminology. When I conduct that experiment, I can then compare those two groups. Let's say my group that ate Oreos for breakfast, after 2 weeks of having Oreos for breakfast every day, but everything else throughout the day was the same as the control group who didn't have Oreos, who had, let's say, Wheaties for breakfast every day. Now I can compare their test grades after 3 weeks. In that comparison, I'm going to gather my data. What are their test grades? Everything else was the same. So I'm comparing only the impact that these Oreos had on their test grades. And based on that analysis, I'm going to draw a conclusion. So let's say my Oreo group all finished with 75s on their test. That's the average for all of them, for those 10 students. But the average for the other group was 78. Not a huge difference, but I can draw a conclusion that no, Oreos did not help. Because what would have happened anyway without the Oreos is I would have scored a little bit better. Maybe both groups get a 75. And I'd say, well, Oreos didn't have any impact on the outcome. So I have gathered evidence that disproves my hypothesis. So one of the important things about the scientific method is we gather evidence that either disproves my hypothesis. If the grades are different and the Oreos are lower, then I have disproven my hypothesis. If the grades are the same, I've disproven my hypothesis because I proposed the test grades would increase. If they do indeed increase, I'm not going to say I proved my hypothesis, but that I gathered evidence supporting it, that the evidence supports my hypothesis. The reason we don't say I proved my hypothesis is because of those controlled variables. I can brainstorm and think of everything I think that might possibly affect test grades, but do I know I've covered everything? Maybe there's some difference I didn't account for. So until I can be absolutely sure that I have accounted for every possible other variable, I can't really say I proved my hypothesis, only that the evidence supports it. So then what scientists will do is continue to study a similar hypothesis, and they will expand and they will test this in lots of different permutations, and they will look at different controlled variables. And the more evidence they gather, the more times they test this, the more evidence that supports the hypothesis, the stronger, the more confident we are that it is correct. But we don't say it's proven until we've been able to test every single possible variable. It's really not possible. So that gets to my next point here on the scientific method is some definitions, hypotheses versus theories. So we'll get laws out of the way first. Scientific laws are physical laws, laws that govern the universe are things that we've found to be universal over and over and over and over again. The law of gravity. We know how it works. We know that number works. We know pretty much it's going to apply everywhere. Do we understand the mechanisms underneath it? Not always. But we know it works this way all the time. So hypotheses and theories, these are really the two I want to focus on. A hypothesis comes at the beginning of your scientific method, before you've gathered any evidence. I have an opinion. It's based on experience. It might be based on doing a literature review. I can go to the library. I can Google, has anybody else looked at this and what did they find out? all right, here's what I think is going to happen based on those experiences, based on my background research. But I have not personally gathered any evidence. I do not have evidence yet. So it's still a hypothesis. So a hypothesis comes at the start of an experiment and is not backed by evidence. Most times when we hear the term theory used colloquially in casual conversation, it's being misused. And what's meant is, here's my hypothesis. Here's what I propose the outcome's going to be. Now let's test that and see what happens. Scientifically, theories mean something very different. A theory is formed, it's a working model. It's a model that has been developed that has predictive capability that I can use to explain phenomenon in the natural world. And when I go out and look in the natural world at new and different things, they work the way my model predicts, because I have lots and lots and lots and lots of evidence to support them. So a theory is built out of lots and lots of rigorous testing of many related hypotheses. So back to our Oreo example, Oreo cookies aren't the only thing that can increase my test grades. So if I want a theory about how to maximize my grades in school, it wouldn't just be about Oreos. It would be about the best things to eat at the best time, the best way to study, study approaches. I would propose hypotheses about all of these things. I would test all of them to have evidence supporting all of these different factors. And then I could come up with a broad statement, a theory that's predictive. So a theory gives me the answer because it's based on evidence that's been accumulated over a broad range. So I could have a theory of how to optimize my grades. And that would include, if I get 7 to 8 hours of sleep every night and spend 2 hours every day studying, broken up into smaller study sessions, and I eat a healthy diet, minimizing how much sugar I have, and I read the book as well as attend lectures, and I attend those lectures, and I choose the best instructors, or instructors that use the most varied methods to teach and base their teaching on best practices, then I will have the best grades possible. All right, that's my predictive model. I can go out in the world and I can look and see. Let me see the best performing students. Do they follow those? Oh, look at that. They do. How about that? How about if I change that up? Then what happens? So a theory is predictive because it's based on all of the evidence we've already accumulated. So when you hear theories a lot of times used, especially on TV, they're misusing it, and what they mean is hypothesis. If they're proposing what the outcome will be before they have evidence, it's a hypothesis. So if we look at the strengths and limitations of the scientific method, a lot of people, until you're really familiar with how it works, till you've seen it work, like we'll see in lab, don't understand that idea of never saying that we proved something, only that we've gathered evidence for it. We can disprove it. All we need is one time for it to fail, and we've disproved our hypothesis. But by not saying that I've proved it, only gathered evidence, that's one of the biggest strengths of the scientific method. And the reason for that is the scientific method never stops inquiry. I never say, if I say I've proven something, I'm not going to look any further. All right, so it's proven. I know it. All right, I'm not going to look any further. Scientists just say they've accumulated evidence. The strength of that is that as new information comes to light, as new tools and technologies develop, as we're able to look at things in different ways, as we're able to test things in a different setting, under different circumstances, we identify a variable that we never even considered before that might have an impact. We're going to continue to test, and we're going to keep looking at those same things until we have a better and better and better and better picture. And then we're going to keep testing them more. So science is considered progressive because we continuously build and improve our knowledge. So we progress and build. We don't prove conclusively because it's a big universe. We'd have to test every single variable. And there are an infinite number of variables, some that we don't even know about at any given time. So we keep that open so we can always go back and look and test again. And that's a strength because the more times we try to prove something is wrong, which is what our scientific method is trying to do, because we can prove it's wrong, we can only gather evidence that we're right. So we always try to prove that it's wrong, and the more times and the more different ways we try to prove that it's wrong and we can't, the stronger our evidence becomes, the stronger our foundation for how we know something works, the stronger that becomes. The limitations are we do have to have observable and measurable phenomenon. Sometimes something might be observable, but we don't have the instrumentation to measure it yet. So there are things that we can't study in physics. A lot of times they're ahead of the curve. They're thinking ahead of what will happen before we have the ability, before we had the Hubble Space Telescope. There were things that we couldn't study. So sometimes our hypotheses are proposed and worked out mathematically until we can actually go out and gather physical evidence. There are also many phenomena that can't be studied through the scientific method. Art, for example. I can look at art, I can measure art, how much color was used, what the pigmentations were, what makes good art. So subjective things like what's good, what's bad, taste in food, what food's better. That's very subjective. It's different for different people and our different experiences, the culture we grew up in, the foods that we grow up with make us appreciate different foods. So anything that's based on belief and feeling, and what's a preference? That is subjective. So what's the best art? What's the best music? Well, that's very subjective. So we can't really, there are parts of music we can study with science, there are parts of art we can study with science. But that subjective portion of what do you think is the best music? Subjective. So there are limitations. A lot of fields that do deal in qualitative data will apply a modified version of the scientific method and try to gather quantitative data on those qualitative aspects. Like how many people prefer rap music to country music. I can gather quantitative data, but again, the basis, it's really qualitative. It's subjective. So there are many things. It doesn't mean that they aren't valid areas of study. It doesn't mean they aren't valid beliefs, but it means they are not in the realm of the natural sciences. They fall in another realm: history, art, music, literature, theology, philosophy. They fall into another realm, into another discipline. So we can try to apply the scientific method only because it gives us a nice structured, methodical way to approach and answer questions, but it just doesn't fall into that discipline. Doesn't mean any of those areas are not valid, only that they're not science. All right, I think that gets us through Chapter 1. Go ahead, follow through on those, on the study guide, on our learning objectives, and I will get our next slideshow posted soon.